Hello and a warm welcome to our Cenibra podcast, which we named Looking at Science, relating broadly to our product lines, looking at cells and looking at layers. My name is Christoph Enz, and in this series we engage with customers, suppliers, friends and business partners and talk about science and things around it, in a very general sense. We will meet with interesting people who talk about their work, their research and their experience with us, our technologies or our common playground in science. It can be quite specific, but we will also dive into broader aspects of scientific work and topics related to it. We're doing this not regularly, but in a very opportunistic way. So when we run into an interesting topic and conversation partner and all of us can squeeze it in, you will find something in this corner of our website. New episodes will be announced on our social media, so no need to subscribe here. If you like it, just enjoy and pop by again. And if you don't, please tell us so we can learn.
Hi and welcome again. My name is Thomas and I'm here once more with Lisa Bodily of the Face Holographic Imaging team. Last episode, Lisa has given us a fantastic overview of some of the features, potential applications, as well as successful use of the HoloMonitor. However, so far we have only scratched the surface and have not talked about technical details or have taken a closer look at specific applications. Just as in the last episode, I have prepared a few more questions for you, and I'm really happy that you are here to face them. Welcome, Lisa.
Thank you, Thomas, for welcoming me back. Let's do this.
So, Lisa, last episode, we wrapped up talking about the presentation of already acquired images. Now, I would like to take one step back and understand how we got to this point. I previously mentioned the highly compact design of the HoloMonitor, which means you can capture images right in your incubator. How does this quantitative face imaging work exactly?
Yes, so how does quantitative face imaging work? So let's say quantitative face imaging, short, QPI from now on. So let's save us some time here. It will be a bit interesting for me trying to describe it on a podcast rather than showing it in real life. But let's see if I'm able here as well to describe it in words, right? So human cells are transparent, which means they neither absorb nor scatter light, which makes them difficult to see under a traditional microscope, such as, the cube of party eyes we have in water glasses, the type of clear eyes that we never get or never manage to make at home. So cells delay light waves as they pass through them in the same way shallow water around a sandbar slows down ocean waves. And PHI's microscope technology measures this delay and makes it possible to image and measure unaffected cells without this genetic manipulation or toxic staining used by conventional microscopy or cell analysis methods. Light creates a wave pattern like water waves when two light beams meet. So in our HoloMonitor system, a low-power 635 nanometer red coherent laser or laser beam is split into two in the system, and one beam is passing through the cells that are placed in their vessel on the stage of the HoloMonitor, while the other beam passes by the back of the system by the sample as a reference beam. So we have a sample beam and a reference beam. When these two beams meet again, a wave pattern, a so-called hologram, is formed, which contains information about how much that laser light was delayed when it passed through the cells, through your cell sample. That hologram is then photographed with a digital image sensor. And from the information in the hologram, a three-dimensional image of the cells is then computer calculated, where the height and color of the cells reflect the delay of the light. So this holographic microscopy technology makes it possible to measure cell volume and other important properties which cannot be measured with any other conventional microscopy technology. And the delay causes the light waves to shift relative to each other. And PGI's holographic imaging technology images living cells through this phase shift. Therefore, the company name, Phase Holographic Imaging.
I can imagine that was rather difficult, but you did
Yes.
great. Let's hope all listeners could follow. Is there anything I need to be aware of, and how do I get my pictures and videos to finally show up on my screen?
Yes, maybe more on a note back to the technology. We have a couple of webinars on that. So if audio wasn't enough, then I hope our listeners can check out those technology webinars.
Great.
But yeah, so anything you need to be aware of, and how do you get your pictures? So to note about this technology is that it does not require any labels, any cell labels, any markers, any stains. What the technology, however, needs is a reference for all the quantitative cell measurements. So that is a no cell, zero background. Also, as you can imagine, there is a limit to cell size. And there's actually a great Swedish word called lagom that means not too large and not too small, just right. It's a great Swedish word, lagom. And HoloMonitor is designed to image best adherent growing 2D monolayer mammalian cell cultures. So bacteria, as you know, they're way too small in comparison. And on the other hand, this is not a great fit for imaging large organized spheroids or imaging in vivo. Their optical thickness would just create a huge spike signal. This is way too thick. However, customers of us are studying the formation of spheroids or using thin layers of gels or coatings like hydrogel or matrigel in their setup. And what do you need to get your images on your screen? That would be the HoloMonitor LiveCell imaging software called AppSuite. And with the help of this software and the help of sophisticated computer algorithms, your connected computer will calculate and reconstruct the images into what we see when we're looking at them on the computer screen. So we can look at our cells and our cell images in a grayscale, or as it is digital, in artificial colors, so making science more colorful. And you can also look at your cells in sort of 2D from the top, or with a button click switch to a 3D view that you can also zoom in and out. So if you want to have a certain angle, a certain view onto your cells. And if you want to watch, Thomas, if you want to watch any more cool cell videos, live cell videos, you should check out our YouTube channel.
I did, so maybe the listeners should do. But thank you, Lisa, for this illustrative explanation. That sounds quite easy. But is there anything else I should consider? What kind of sample holders can be used, and are there any specific requirements or consumables?
All right, so no special vessel formats as a first one. So we go from 35 millimeter Petri dish to cell counting slides, micro slides, 6 well plates, 24 well plates, or 96 well plates from various manufacturers. However, as a disclaimer here, we have a list of thoroughly tested vessels that we test for their optical imaging quality here in-house, and we're always expanding on that list as well. Those tested vessels have also their own easy setup template in the software to make it much more faster and easier to use them. So that was about the vessel formats. Let's have a look at, all right, like motorized stage is the other part. So you can click your microplate. So you can either click one microplate, 4 Petri dishes, or 3 slides to run at the same experiment, and click those onto the motorized stage. And then the stage moves from well to well, and you can capture multiple positions within each well. And there's no downtime in between. So like the stage is moving in X, Y, and Z direction, and you could image multiple positions in each well. And on the side of consumables, I can note here our so-called HoloLids that we have developed. They improve image quality, and you can add them. Oh, now I'm trying to describe that as well, the podcast. Do you add them into or onto your plates or onto your Petri dishes? And they will dip into the media. And by doing so, reduce the surface vibration in the media and remove condensation. So we also have developed them so they fit our recommended vessel formats of plates and dishes. And I definitely would recommend using those. You can clean and reuse them as well after your experiment.
And let's be honest, that last point is really great and important for a lot of customers.
Absolutely.
So now let's imagine we have set everything up and we would like to follow cell differentiation and morphology changes over time. Time-lapse imaging always means the generation of a huge volume of data, which can be a bit overwhelming, of course. So how does the HoloMonitor handle data generated during live cell experiments, and how are they displayed in the end?
Yes, we all love data, don't we? So let's have a look. So during each live cell experiment, you can see the images that are being taken immediately in the software in a systematic way. We have a image tree, an interactive vessel map, so you can check out different positions. And the computer is always connected to the system in the incubator via a USB and receives and reconstructs the images on the go, calculates, and then presents the data directly in the software. So I think that's sort of generic. But if we go more into the different assays we have, so we have our guided kinetic assays. And in these assays, you choose what type of result you're after at the end, when you're setting up. And the images are analyzed during the image capturing, and you will get ready graphs also ready during the duration of your experiment. So these assays provide the results on a population level and include proliferation, motility, and dose response. We also have a wound healing assay where you get population level data, but that data is analyzed after the experiment is finished. Now, if you are more interested in following, for example, cell differentiation or morphology changes over time, you would use the in-depth assays, in which you get data on a single cell level, including single cell tracking and cell morphology. So single cell tracking and single cell morphology. And let's have a look at these two apps maybe. So in the morphology app, you will find next to the images of your segmented cells, scatter plots with gating functionality, which many of us are used to from, for example, flow cytometry. And that will make it easy for you to visualize your kinetic cell morphology data. So you can plot also over 30 different morphological features, X, Y, against each other, and investigate your cell sample. That goes for area, thickness, volume, texture, and irregularity of your cells. So you would get, if we sum that up, you get scatter plots in the morphology app that help you visualize the differences between, for example, control and treated cells. And they assist you in comparing cellular characteristic at different time points of the experiment. Second, you have gated regions, which enable you to classify, for example, subpopulations and identify rare cells of interest. In addition, you could add here also to the morphology app the single cell tracking functionality. I will talk more about single cell tracking later. And thirdly, we have histograms that show the cell population distribution of selected morphological parameters at selected time points. So if we switch over to data from the single cell tracking app, well, what data would we get? So we get a cell movement plot. And in that plot, each line would represent how an individual cell has moved over time. It looks a little bit like, a wool ball that a cat would like. This cell movement plots. We have a cell feature plot or cell characteristics plot, where for each cell that is tracked, the morphology change over time can be plotted, like the same way like in morphology. So you can follow area, thickness, volume, texture, you name it. And last but not least, you also have a cell tree plot. So the software automatically detects cell divisions, and the results are displayed in a cell lineage tree plot. So you can follow the mother cells and their cell daughters, and then the whole cell family.
Yeah, that sounds like you have a lot of possibilities. And let's say we stick to one of them, we stick with single cell tracking application. I would not expect the HoloMonitor to generate the highest resolution images, right? But I see its big advantage in acquiring quantitative data on single cell level. Could you explain how the cell analysis is done?
Yeah, you're right, Thomas. HoloMonitor is not like your high resolution microscope, which you would use to study subcellular components. HoloMonitor you use if you wish to study your cell's behavior. Knowing what your cells are up to. What are your cells up to behind closed incubator doors? That thing we all want to know when we are leaving the lab behind for the weekend. What are these guys up to? And it's also not a classical microscope in the traditional way, I would say. But I guess you could say it's a 20 times objective. And each captured field of view is 567 times 567 micrometers. The system's capture rate is up to 1 image per second. Now, that will adjust the more capture positions you choose across your wells, and the motorized stage will move them from each one to the next one. But in the software, it would tell you all this before you start imaging. So how long will one imaging batch take? How many images do you have? What's the size of that data? And the software won't let you start the experiment if you don't have enough storage. So that's good. So you're not running out halfway through. But I guess for us organized researchers, that wouldn't be an issue, keeping our database as well structured. And instead of labeling our cells, we can label our tubes and databases, right? I don't know how you were, but I'm always a very organized person.
Yeah, I think most of us are.
Absolutely. So, however, speaking of resolution, so one of the biggest advantages of using quantitative phase imaging, QPI, is that you receive sharp and precise images of your cells. The cells are identified well separated from the background in comparison, for example, to phase contrast or phase contrast microscopy. That's what I mean. And this advantage allows for precise cell identification and segmentation by the software, which also means that you can get quantitative data on each individual cell in the image and not only the cell population level. And the software automatically identifies all those cells in the image. So the algorithm takes into consideration the cell thickness, the size, and morphology to first separate the cells from the background, second, split adjacent cells, and third, fine-tune the results. So the algorithm identifies cells by setting a threshold that determines what is cell and what is background. And each identified cell is then marked with a dot. So as a user, you can also make adjustments for the background threshold, the sensitivity or object size. And this way you can adjust the shown mask and how it fits your cell segmentation best. You can also make manual changes, for example, removing a cell that you want to exclude from your analysis. There's different segmentation adjustments you can take the software automatically does its job, but it's important for the subsequent analysis to verify that the software algorithm was correctly applied. So, and this is a step you should never miss, no matter what tool or imaging method you are using.
Yes, you're definitely right. Also based on other microscopes and their software, I know that the cell recognition and segmentation can be really challenging. And at certain points, you always have to live with losing a few cells. But are there also any limitations or challenges with the HoloMonitor you can tell or you're willing to tell us about?
Yeah, so we get often questions regarding confluence or thickness of cells or edge cells. So maybe we can talk a bit about that. And those are also typical questions that our application team is helping with. So confluency of cells plays a role for QPI technology. If you have a highly confluent sample with no detectable background, no cell-free area in your image. So if you have that confluent image, how is your software going to know what is the baseline, the zero for any quantitative analysis? That is, for example, a limiting factor. So on the other hand, however, if you have too few cells, you might have inadequate results due that the autofocus is not able to properly focus on the cells. So it's not enough in the image. But I guess that goes for many other imaging techniques as well that are using automated software. If you're running a multi-day experiment, I would say you seed your cells with about maybe 5% confluency and then let them attach for up to 24 hours. Add your treatment, if you have any, and then place your sample on the HoloMonitor. That should be fine. And yeah, cells come in all shapes and sizes, right? The thickest individual cells have been around 25 to 30 micrometer when rounded up for mitosis. Average cells are 10 to 20 when rounded up, usually more like 6 to 9 when flat. And T cells change, but they start somewhere between 5 to 7 micrometers. And if a cell is only half in, half out of the field of view, I would exclude it from the analysis. So because you're not not know 100% how the cell looks like on the other side of the field of view. But in the software in cell identification, you can check a box to include cells or exclude cells that are touching the edge of the image frame.
And it's really nice that you thought of this and that one also has the option to manually exclude the cells. And on the other side, cell differentiation and segmentation has for many people been the easiest by fluorescence signals. So now I have seen that you also offer a green fluorescence module for the HoloMonitor. Would you like to tell me a little bit more about how it can be equipped and for what it can be used in the end?
Yes, Thomas, you've seen that right. We have now a green fluorescence add-on to the HoloMonitor. And if you just use HoloMonitor as its base system, you're going to use the label-free QPI imaging part, where you don't need fluorescence to identify your cells in the image or follow and track them over time. You would do that with the QPI label-free holography channel. However, our customers gave us the feedback that they wanted to broaden the system's application. And that's why we introduced the fluorescence add-on. So in addition to all the label-free holography cell features to study your cell's behavior, you can now also analyze the cell's fluorescence signal in the single cell tracking and the cell morphology assay with HoloMonitor. So you could study cell death using label-free morphology and cell tracking, and then check with the fluorescence add-on for example, when a cell death marker is expressed. You could get more details in co-culture setups with the two channels, holography plus fluorescence. You can have further applications within reporter gene expressions, uptake assays, transfection efficiency, or checking for live cell staining success. And the benefits of this fluorescence add-on module is that it connects just below the HoloMonitor, also in the incubator. So you get then the combined data of the holography and the green fluorescence channel in one experiment, while keeping the light exposure on your cells to analyze them to a minimum, and therefore reducing the photocytotoxicity. And that allows us, I guess, to still do the long-term single cell and population experiments with your cells. And you keep that small footprint in the incubator, and you keep that same intuitive and continued workflow from setup to analysis. So it's a truly integrated add-on module.
It's fantastic to hear that you react to the feedback. Are there any other adaptations based on the specific needs of your customers?
Absolutely. We're always very happy to have a dialogue with our users to see how we can push the QPI limits and its many different applications in cell research. For example, our latest software update brought advanced image analysis features in the guided assay and their user interface. So that was simplifying experiment analysis, and all that feedback came directly from customers. We added the EBD microslide vessel template in the software to make chemotaxis experiments easier and more reproducible to set up. So that's fully integrated. The same way goes actually also for the wound healing assay, that we have a vessel template with the EBD inserts. And also the latest software had some additions in the cell morphology app. That was the CSV export, and further advanced exports for full frames, individual cells, masks, etc. People just wish to export all different things, so our software team made that happen. You see, we continuously make improvements, small and large, and have our system meet the needs of biologists today and tomorrow.
What a nice and a fitting statement to come to an end of this episode. But as kind of an outlook, Lisa, let me ask you one last question. Your team is very engaged in continuously offering a cutting-edge product. I see that PHI is also developing its quantitative phase imaging, or we wanted to stick with QPI, technology for the field of regenerative medicine. So what are your current projects there?
Exactly. So our team focuses on developing our technology for cell-friendly quality control in the regenerative medicine cell manufacturing process. So we see our technology addressing a central puzzle piece for scaling and standardization and making cell-based therapies accessible, affordable, and patient-safe. So, for example, in our ongoing smart manufacturing project, we are setting up a mesenchymal stem cell manufacturing chain that is suitable for point-of-care production, including data collection, analysis, and storage. And we do that together with our partners, that is QIAGEN, Analytics Leader, SAS, Biospherics, and we do that at the Wake Forest Institute for Regenerative Medicine in North Carolina. On the other hand, we also explore further applications with our many academic collaborators and partners. For example, Dr. Robert Judson Torres and his lab at the Huntsman Cancer Institute at the University of Utah, where we're developing the use of QPI in precision medicine.
Impressive. Thank you so much, Lisa, also again for being here once more and giving us this detailed overview. So if our listeners have only listened to this episode, how can people reach out to you?
You're welcome, Thomas. So to contact our team, you can write us at info@phiab.com or visit our website, also phiab.com. Thank you so much for having me.
And thank you for being here. And thanks also to all of our listeners for joining us. And you can hear us the next time in our Looking at Cells podcast. In the meantime, and if you have any questions, feel free to reach out to one of our specialists and visit our website at cenibra.de.
This was Cenibra's Looking at Science podcast. Thanks for listening, and for any feedback, please send us a note. And if you follow us on social media, you'll be alerted about the next issue. All the best, and talk soon.